The review of pertinent aspects from biology and physiology was important as a background of what may happen and specifically not happen when implant materials and devices are introduced inside the body. That particular introduction creates an interface between the biological physiological environment, which contains cells, extracellular matrix, biochemical and biophysical stimuli, blood vessels and nerves, and on the other side, when an implant is present at that particular location, we do have the cells, perhaps the biophysical and biochemical stimuli, blood vessels and nerves, but in the middle part, the physiological extracellular matrix is now replaced by the synthetic biomaterial, which may be a ceramic, a metal, a plastic, a composite of two or more of the previously mentioned types of materials. This approach so far required our in some detailed review of the tissue wound healing process, all the pertinent stages, and in addition from biology, how proteins absorb on substrates and how cells interact with the adsorbed proteins and therefore interact with the substrate surfaces. And those substrates could be either naturally occurring or synthetic materials. Recent advances in biomaterials made possible an alternative approach, and that approach now starts with the synthetic materials out of which devices, implantable devices, are composed, and searches how these particular novel materials interact in the biological environment, which of course contains biological fluids, proteins, cells, and tissues. These novel materials are called either nanostructure materials or nanophase materials or nanomaterials. The topic of lecture 18 is cell interactions with nanostructure materials. We'll start with a definition of nanostructured or nanomaterials. These are synthetic materials which are insoluble, and they are characterized by having at least one external dimension or an internal structure which is on the scale from 1 to 100 nanometers. The important requirement for medical applications of nanostructural materials is the following. Such biomaterials, like all the other implant biomaterials, are introduced inside the bodies of humans and mammals, and they are expected to function safely and successfully achieve the specific objectives for various periods of time, and most probably for long periods of time, specifically the duration of the life of the recipient patient. A very important and new aspect regarding nanostructural materials is that of the specific dimension, which sometimes it's also referred as atomic domain or grain size. An illustration which I hope will help us visualize this particular process is that of nanostructured ceramics. As shown in the schematic frame A, we have crystal nucleation which comes about to form a solid material. You can see the grains growing, shown schematically in frame B. And eventually we have all of these coalescing and bringing about all these particular grains together. So If we use this particular illustration and then go back to rephrase the definition of a nanophase nanostructure material, the important thing to keep in mind is that these materials have atomic domains, so-called grain sizes, which are less than 100 nanometers in at least one direction. In contrast, the conventional or so-called micron size are the materials which have grain sizes greater than 100 nanometers. Another very important aspect to keep in mind when we compare the nanostructure versus the conventional material, both materials have the same exactly chemical composition. The only thing that is different is this aspect of a dimension of the grain size. The nanostructure materials, they must have a dimension of less than 100 nanometers in at least one direction. Otherwise, the materials are classified as conventional or micron size. The excitement regarding nanostructure materials was their potential to promote functions from select mammalian cells which are pertinent to new tissue formation. Applications of nanostructure biomaterials have been investigated in terms of prosthetic devices, in tissue engineering, tissue regeneration, and several other fields. Biomedical engineers interested in implantable materials and devices investigated the use of nanostructure materials from the specific point of how the injured tissue around these implants heals and how new tissue is formed around nanostructure materials and devices. This approach, of course, led to investigation of protein adsorption on nanostructure materials and subsequent interaction of cells with the adsorbed materials. I will present to you the results of some of the very first research which was conducted in this area. It happened to be in my laboratory, and the experimental conditions were as follows. We use nanophase and compare them to conventional alumina, titanium, and hydroxylapatite. As you know, hydroxylapatite is a ceramic, but in addition, alumina and titanium are ceramics. Specifically, they are the oxidized layer which is formed on the top of aluminum and titanium, aluminum and titanium being metals. The oxidized layer which is formed on the top of these respective metals when these metals are exposed in an environment that contains oxygen. The layer on the top of the oxidized layer is, of course, ceramic. The studies used, of course, bone cells. We've been interested in the applications of implants in bone, and as a comparison, we use some other cells in the laboratory, of course, the ubiquitous fibroblasts, which are considered a competing cell line because fibroblasts are associated with the formation of soft tissues. In contrast, osteoblasts are associated with the production of bone, a hard tissue. In addition, we use endothelial cells as a third cell line in order to see what various cell lines do in conjunction with nanostructure materials, how they interact with those materials. Cell culture conditions were standard cell culture conditions with the appropriate media and in a humidified 37 degrees centigrade 5% CO2 balanced air environment for various periods of time pertinent to the function of the cells which was under investigation. Since cell adhesion is the crucial function for the survival of anchorage-dependent cells, cell adhesion on nanostructured materials was the first one to be investigated in these studies. The results of osteoblast adhesion on various ceramics, specifically alumina, titania, and hydroxyl apatite, is shown on this slide. Please notice that in each case, by that I mean the case of each ceramic, we have a substrate which has grain sizes above 100, therefore it is the conventional, and two other substrates which have different grain sizes, one much smaller than the other. As you notice, the adhesion of osteoblasts on either nanostructured alumina or titania or hydroxylapatite compared to that of these cells on their respective conventional substrates was enhanced in the case of nanostructured substrates. This was an exciting result. However, some people will say, well, bone cells, the osteoblasts, like ceramics because ceramic is a major component of the native bone. However, there were some additional results which continued our excitement about this particular study. The results which added to the excitement of this particular study were those related to the specificity of the interactions of only certain cells and not every cell with the nanostructure ceramics. These results are illustrated on this particular slide. You see here the adhesive interactions of three types of cells: osteoblasts, fibroblasts, and endothelial cells. In each case, the blue bars are those of the results of adhesion of the respective cells on conventional, that is now substrates with grain sizes larger than 100 nanometers. The other two bars represent the adhesive interaction of osteoblasts, fibroblasts, and endothelial cells, respectively on two sizes of nanostructural materials, those with grain sizes of 45 nanometers, those are the green bars, and those with grain sizes of 24 nanometers grain sizes, and those results are represented by the red bars. Let us review the results of each one cell separately. What we see in the case of the osteoblast is that these cells interact adhesively with nanostructural materials independent of the grain size, and that of course result is enhanced in comparison to adhesion of osteoblasts on conventional ceramics. The interesting thing is that there is some increase of that adhesion as the size of the grain size is smaller. In contrast, adhesion of fibroblasts on the nanostructured ceramics compared to that on the conventional ceramics decreases as the grain size of the ceramic is becoming smaller and smaller. This particular result is of great importance because, as I mentioned earlier, we consider fibroblasts as a competing cell line for the function of new bone formation. Fibroblasts are associated with formation of soft tissue. In contrast, osteoblasts are the ones cells which promote or are the ones which create new tissue, namely bone. In the case of endothelial cells, you see overall very small adhesion on all ceramics. But if you see the trend, again, adhesion of endothelial cells on the nanostructure ceramics is lower than that on the conventional ceramics. This particular result provided very intriguing and for the first time information of the preference of certain cells, specifically osteoblasts on ceramics here. The osteoblast interaction with nanostructure ceramics is enhanced in comparison to what we see when these cells adhere on conventional ceramics. This intriguing result promoted the investigation of the mechanism of cell adhesion on nanostructure ceramics, therefore additional experiments. The results shown on this slide are extremely important. It is a busy slide, but what I would like to point to you again is that the blue bars reflect the adhesion of different cells, specifically osteoblasts, fibroblasts, and endothelial cells. The results are shown on 3 separate graphs. Blue bars, conventional. Green and red bars, 2 different grain sizes, nanostructure, ceramics, specifically alumina. This is a busy slide, but the important message is the following. Independent of whether the cells interacted for half hour, 1, 2, or 4 hours, and independent of whether the osteoblasts and the fibroblasts and the endothelial cells were the cells investigated, the interaction was very low and there was no difference between how the cells interacted or how many cells interacted with the nanostructures versus the conventional ceramics. What was the reason? In this particular case, the experiments took place in the absence of serum proteins. For those of you who are not familiar with the process of culturing cells in the laboratory, when we culture cells, we provide them with what we call serum. And this is the liquid part of blood, usually from cows, after blood was clotted. Therefore, this particular serum contains the liquid part of blood without fibrinogen fibrin. For some reason, culture cells like to have that particular supplement in their supernatant medium. In the absence of serum proteins, we have, as the results shown here, no specific and different interaction of the various cells and of a specific cell on the nanostructure versus the conventional control substrate. I'm repeating the conclusion of these experiments in words on this slide because I want to emphasize the results. In the absence of serum proteins, cell adhesion was very low and similar on the nanophase and the conventional ceramic substrates tested, and for all types of cells tested in this particular study. The result that in the absence of adhesive proteins, the select interaction of specific cells with the substrate material is lost, prompted the investigation of the mechanisms of osteoblast adhesion on nanophase ceramics, focusing on the role of proteins. Additional justification for this study was provided by what is known, specifically that within seconds from the exposure of a biomaterial to either biological fluids or proteins somewhere in the body, proteins will absorb on the surface of that material. Depending on the type of protein, the concentration, the distribution, and the conformation, then these adsorbed proteins will subsequently mediate the adhesive interaction of cells from the surrounding tissues. The results of protein adsorption on conventional, shown by the blue bar, and on nanostructured alumina, shown by the red bar, is shown on this particular figure. The proteins contained in fetal bovine serum, as I mentioned, is a cocktail of all kinds of proteins which are found in the serum of mammals. When now individual proteins, one at a time, were taken and its adsorption was studied on these two types of alumina, keep in mind, only difference in the grain sizes, not in the chemistry. All of these substrates are ceramics alumina. The blue bars represent adsorption of these proteins on conventional alumina. The red bars, adsorption of these proteins on nanostructured alumina. As you can see, in the case of albumin, which is a protein very important for maintaining the osmotic equilibrium in the body, but it's not an adhesive protein, the two bars are of the same height. Therefore, there is no difference between adsorption of albumin on either conventional or nanostructured alumina. In the case of laminin, which is a protein found in the vascular tissue and a protein which mediates the adhesive interaction of endothelial cells, the ones that line the innermost layer of our cardiovascular system, notice that laminin adsorbs preferentially on conventional but much lower on nanostructured alumina. The story is much different when collagen, fibronectin, and vitronectin were investigated. These three proteins are known to mediate adhesive interaction of osteoblasts with substrates, natural and synthetic. And you notice that in all three cases, the red bars of representing the adsorption of collagen, fibronectin, and vitronectin on the nanostructural alumina are much higher than what adsorption was obtained for collagen, fibronectin, and vitronectin on the respective conventional alumina. Adsorption of proteins on biomaterials is important because subsequently cells which adhere on the adsorbed proteins will survive and function. In contrast, the cells which do not have that very important mediation of the adsorbed proteins are not going to survive. This slide summarizes the results obtained when osteoblasts adhere on nanophase, nanostructured alumina, or conventional alumina on which select proteins had been pre-adsorbed. The objective of the study was to investigate whether there any difference of osteoblast adhesion on the nanostructure or the nanophase alumina, and to find out which protein or proteins are responsible for that particular result, which is different on the two substrates. The first experiment used the traditional 10% by volume concentration of serum from cows. This particular liquid part of the blood of cows contains various proteins, not all of the proteins are known and we don't know the concentration, specific concentration of each one of those proteins. Suffice to say that this is a standard volume of serum used in culturing cells. On this graph, the blue bars illustrate the results obtained when osteoblasts adhere on the conventional ceramics. The red bars represent the results obtained when osteoblasts adhere on the nanostructure ceramics. When 10% bovine serum was used, the osteoblasts adhere in larger numbers, as the red bar shows, under 10% FBS. The osteoblasts preferred adhering on the nanostructure alumina. Keep in mind again that this particular concentration is what I may call a cocktail containing various proteins. The rest of the results show what was obtained when we used one protein at a time. First and foremost, albumin. The two bars, blue and red, are of the same height. Albumin is a protein known for its role in maintaining the osmotic equilibrium in the body, but it's not an adhesive protein. So the non-difference in the adhesion of osteoblasts on conventional and nanostructure alumina is not a surprise. Similarly, when laminin was the protein pre-adsorbed, we noticed no difference between adhesion on the nanostructure and the conventional ceramics. Laminin is a protein present in the vascular tissue and therefore important in the adhesive interaction of endothelial cells, but not of osteoblasts. The results which were obtained when osteoblasts adhere on substrates on which we had pre-adsorbed either collagen or fibronectin or vitronectin were totally different. In all three cases, the red bars show enhanced number of osteoblasts adhering on the nanostructure versus the respective conventional substrates. Conventional substrates, again, shown as blue bars. Red bars represent the cell adhesion on the nanostructure substrates. These results provided evidence that certain proteins are involved in mediating the adhesive interaction of osteoblasts on substrates. Specific proteins mediate the adhesive interaction of specific cells of certain tissues in the body. An explanation regarding the underlying mechanism of osteoblast adhesion on nanostructure versus the conventional alumina is shown in this cartoon, which was put together by Tom Webster as part of his doctoral thesis. In this cartoon, you see on the upper row an indication of osteoblasts. On the bottom row, we have the conventional alumina on the left, the nanophase alumina on the right. The mediators of the interaction of the cells with the substrates are, of course, proteins, and in this case, the protein is vitronectin. It is known from literature search that vitronectin is a very specific protein for mediating the osteoblast adhesion. Vitronectin has many epitopes or domains which are composed of specific amino acids which are recognized by the receptors on the membranes of osteoblasts. One of those domains is RGD, arginine glycine aspartic acid. Dr. Webster's explanation was that when vitronectin is adsorbed on conventional alumina, its conformation or the opening up or the particular morphology that the protein takes on that substrate is such that the adhesive domain of RGD is hidden away from the osteoblast. Therefore, the osteoblast cannot interact adhesively very well with the conventional ceramic. In contrast, when vitronectin interacts with the nanophase alumina, the conformation of the protein, this particular spreading out and opening up, exposes the RGD domains and makes it much easier for the coming cell, the osteoblast, to interact with that domain. That perhaps explains why osteoblasts preferentially adhere in larger numbers with nanostructure versus the conventional alumina. I didn't present all the experiments conducted in this particular study, but I would like to reach the conclusion here, and it was that nanoscale features or grain sizes modulate the type, the amount, and the conformation of select proteins, and that of course dictates the absorption of proteins on substrates. This protein interaction subsequently promotes cell functions, first of all adhesion, but subsequent functions as well, and brings about what we call selectivity by specific cells. This is a remarkable feature of the nanostructure materials because this result is obtained by just putting together a different formulation of the biomaterial substrate. The chemistry is the same. The grain sizes are the only aspects which are different. In the other studies which we reviewed, namely the chemical modification substrates, using information from biology and physiology, what are the mechanisms of cell adhering on proteins, those studies require to know what proteins are involved, to know what particular domains on the protein are recognized by the cells, and subsequently taking those proteins, which means now chemical production of those proteins, going through a lot of serious chemistry to immobilize those particular proteins or peptides on substrates, making sure that those substrates remain bioactive through all this chemical processing, and making sure that once the cells come into the picture, the cells recognize those adsorbed proteins or immobilize, I should say, proteins and peptides and interact with them to give us the desirable outcome, specifically either the adhesion or other functions which are related to new tissue formation. Our research activities using nanostructure ceramics continued in another direction which will be summarized in the slides which follow. These studies use polymeric ceramic composites. So we have two types of traditional materials, a polymer part and a ceramic part. The motivation for the research was as follows. We need biomaterials which have properties similar to those of physiological bone. If you look into the physiology of bone, what we have here is a composite material which contains both a polymer, naturally occurring, of course, primarily collagen, and a ceramic, which is hydroxyl apatite. Most importantly is another aspect, namely that the composite material in bone, the naturally occurring bone, has surface grain sizes in the nanometer range. What we may say then is that bone is a nanostructure, naturally occurring material. I will summarize the results obtained from the study in which we use polymer and ceramic composites. The polymer was polylactic acid, a chemical compound which has been used extensively in tissue engineering. For the ceramic composite, we use a nanoceramic, specifically alumina, with grain sizes of 93 nanometers, and we use the microphase or conventional ceramic alumina with grain sizes 167 nanometers. The composites were made by mixing polylactic acid with various weight percentages of alumina, 30, 40, and 50% by weight. The cells used in the study were bone-forming cells, osteoblasts, and for comparison's sake, fibroblasts, the line which is considered as a competitor because fibroblasts form soft tissue, osteoblasts form hard tissue, bone. The cell conditions were standard cell culture conditions, a humidified 37 degrees centigrade temperature, 5% CO2, balanced air, and Dulbecko's modified Eagles medium and 12% fetal bovine serum, standard cell culture conditions for the osteoblasts used in the study. This slide summarizes the results which were obtained when either fibroblasts or osteoblasts were allowed to adhere on either nanostructure alumina polylactic acid composites. In both phases or frames, I should say, of this slide, we have the red bars which represent the results with the osteoblasts and the yellow bars which represent the results with fibroblasts. You notice that on the y-axis we have the number of cells which were adhering on the respective substrates, and the substrate composition was 0% alumina, which means 100% polymer, then by weight 30%, 40, 50%, and then 100% alumina, which means 100% a ceramic. When either one of those two cells tested, either the fibroblasts or the osteoblasts by themselves, were allowed to adhere on the substrates tested, you notice that there was no difference in the results. You may say that either one type of cell adheres similarly on all the substrates tested, independent of the respective composition. In the case of nanophase alumina, however, there were some very interesting and different results. On the adhesion on the substrate, which is 100% a polymer, that means those are the two bars over 0%, you notice that both fibroblasts and osteoblasts adhere in a similar manner. As the composition of the ceramic, however, increased from 30 to 40 to 50%, all the way to 100% ceramic, you notice that the fibroblasts, the yellow bars, remained relatively low and pretty much similar. However, the red bars, which reflect the adhesion of osteoblasts on the respective substrates, increase with the increasing concentration of the ceramic, and it was maximum on the 100% nanophase alumina. Another important aspect of this study was determination of the mechanical properties of the substrates. For that purpose, the bending moduli of these preparations were tested. The results are shown on this table. On the very extreme left column, you see the results of pure polylactic acid, a polymer, in the order of 300. On the extreme right-hand column, you see the results of pure ceramic, 100% ceramic, which is at least by a factor of 10 lower. Ceramics are known to be very fragile, independent of whether they're nanophase or conventional alumina. In the middle part, you see the results of the bending moduli for the three composites tested, specifically 30%, 40%, 50% by weight ceramic. In the case of the conventional alumina, the modulus increased from 700 to 750 as the respective alumina composition was increased from 30% to 40% to 50%. In contrast, and very interestingly, the nanophase alumina composites with polylactic acid exhibited increased bending modulus as the respective composition in the nanophase alumina increased from 30 to 40 to 50%. In the case of the 50%, the modulus was in the order of 3,500. Definitely, the bending modulus of bones in the human body is different depending on what part of the skeleton the bone comes from. But if we take one example, let's say the human femur, it has a bending modulus of 1,900. The important conclusion here is to notice that by putting together a synthetic composite that imitates not only the relative composition of real bone, namely having a polymeric part and a ceramic part, but by increasing the content of the nanophase ceramic, we reach a bending modulus which is in the order of magnitude of what is found in some parts of the human bone skeleton. In summarizing the results of this study, composite materials which contain a nanophase nanostructure component exhibit some very desirable characteristics which are comparable to what we see in respective tissues in the body. Specifically, they promote interactions of specific cells from surrounding tissues. The example that I illustrated is the select interaction of osteoblasts but not of fibroblasts. And in addition, these composites exhibit desirable mechanical properties when compared to those of the surrounding tissues. For example, in this particular case, bone. The early excitement regarding the potential of nanostructure materials for biomedical application has been tempered a little bit in the interim, because now researchers and the general public have become aware of certain aspects which may be unique to this type of novel biomaterials. The present challenges regarding nanostructure materials are related to safety of these novel biomaterials. I will use an example among those of my personal research interest, and it relates, of course, to bone and nanostructure materials which are considered for applications in bone. By the end of the teenage years, the human skeleton does not grow anymore in the sense of achieving extra height. That, however, does not mean that adults have, for the rest of their lives, the same bone that they had at the time when they were 18 or 17 years old. What happens in adulthood is the following. There are certain cells in bone called osteoblasts which are responsible in forming new bone. In addition, in bone, there are a different type of cells called osteoclasts which break down and absorb bone. The two histology micrographs shown on this slide illustrate osteoblasts on the left frame You can see at the bottom of this particular left-hand photograph the hard tissue bone. On the top of it is a series of little oval-looking entities which are the osteoblasts. On the right-hand photograph of this slide, you see the osteoclasts in action. Notice that the osteoclasts are bigger cells, and in addition they have many more nuclei, those dark dots inside each of these cells. These cells have the capacity of attaching tenaciously on old bone, and once they secure the perimeter of their cytoplasm by very strong adhesion, they bombard the tissue underneath, the old bone tissue underneath, with all kinds of chemical compounds. Some of those chemical compounds have the ability to break down the organic phase of bone, which is composed primarily of collagen and a few other collagenous and non-collagenous proteins. In addition, some of the chemical compounds which the active osteoclasts release have the capability of breaking down the inorganic phase of bone, which is of course primarily hydroxylapatite. As a result of that, the old bone is broken down and removed, and the osteoblasts come at the end of that particular action of the osteoclast and start building and depositing new bone. This particular process is a balanced process in the body and assures renewal of the bone structure and the skeleton in adult humans for the remaining of their lives. Resorption of the extracellular matrix of bone by activated osteoclasts can be checked in the laboratory by doing a pertinent experiment. The results of such an experiment are shown on this slide. On the scanning electron micrograph on the left, you have an illustration of the extracellular matrix of bone. On the scanning electron micrograph on the right, you see the results of what happens once osteoclasts are allowed to adhere and do their function, namely resorb the extracellular matrix of bone. You can see these darker areas which are the pits of the removed extracellular matrix of bone, which have resulted from the action and function of the activated osteoclasts. In some respect, one may say that this particular action of osteoclast renders the extracellular matrix of bone as a degradable material. And the question is, what happens to those little pieces of extracellular matrix of this degradable bone? Where do they go in the body, and what particular action or effect they may have in the parts of the body they accumulate? This question, when the time came to consider nanostructure implant biomaterials, became a question of biosafety, to be or not to be biodegradable. Because as a result of the action of osteoclasts, any naturally occurring or synthetic material which is going to be replacing bone is going to be degraded. Some of these aspects can be tested in the laboratory by using cultured osteoclasts. The results of a pertinent study are shown on this slide. We have two types of substrates here. On the top row, we have alumina. On the left scanning electron micrograph, you see the results of the removal, the resorption of the nanophase alumina from a substrate by activated osteoclast, as evidenced by the big dark area. The nanophase alumina have been resorbed by the activated osteoblast. That particular area is much, much bigger than the fewer and smaller areas which are on the conventional alumina. The pertinent scanning electron micrograph is the one on the top row right hand side. A similar result was obtained when, instead of alumina, hydroxylapatite was used as a substrate. The results are shown on the two scanning electron micrographs on the lower row. On the left-hand side, low row, the nanophase hydroxylapatite has been resorbed, and the area, as you can see, is now devoid of the substrate. This was the outcome of the activated osteoclast function. In comparison, on the conventional hydroxylapatite, the scanning electron micrograph on the lower row right-hand side, those areas of the resorption are fewer and much more smaller and more dispersed on the surface of the substrate. Release of particles and molecules from implanted materials and devices in the human body have always raised questions and concerns regarding potential health and safety issues. These aspects are also of importance in the case of nanostructured materials, especially when we take into consideration the dimension of biological systems. We have seen this particular slide before, which gives us a range of dimensions. A mammalian cell has a characteristic dimension of 100 micrometers, a bacterium of 10 micrometers. But when we get down to the viruses with characteristic dimensions of 100 nanometers and small molecules in the order of 1 nanometer, we have a special situation which is encountered only in the case, potentially the case of nanostructured materials. Because when we take into consideration the characteristic grain size of nanostructured materials, less than 100 nanometers, these are the right size to interact with cells and are structured to do so. This slide illustrates in a cartoon fashion the concerns expressed by our colleagues in the medical health profession regarding the potential diseases that may arise as a result to, first, the type of nanoparticles and the concentration of nanoparticles in various parts of the human body. One of the first questions was, where do these nanoparticles accumulate inside the cells? Do they go to the nucleus? Do they go to the mitochondria? Do they concentrate in the cytoplasm, or they get just stuck on the membranes? Once they are found inside the cells, then they can be distributed in various parts of the body. For example, the gastrointestinal system, then the skeleton, brain, and maybe they're going to cause all kinds of diseases there, lungs, circulation, heart, skin, and other organs. The distribution of nanoparticles in various parts of the body and their accumulation, their concentration, has been of major concern, and this has become an issue which needs to be both investigated and it has to be answered in definitive ways to prove that the nanostructure materials and any nanoparticles that may be shed from them are safe in the body of humans. Biomedical engineers have dealt and addressed the biosafety concerns regarding nanostructure materials from various perspectives. I will summarize some of the important concerns and results of these studies in the slides that follow. The safety concerns cover a wide range of issues. First and foremost, are the nanostructure materials and the nanostructure particles stable inside the human body in the environment of physiology? The type, the size and amount and distribution of the degradation products is another major issue in vivo. Are the effects of the degraded material products detrimental on cell functions pertinent to new tissue formation? This particular aspect will be of great importance to biomedical materials engineers who are interested in developing novel materials for implant devices. Last but not least, toxicity, which means release of chemical compounds. Independent of their number, even in small concentrations, some of those released compounds may be toxic and therefore kill cells and tissues, not only locally, but if they're distributed in the whole body, that is systemically, they may cause all kinds of other problems in parts of the body far away from the site of the implant nanostructure material and of the site where these particles have been produced. Concerns at the cellular and tissue level include the effects of toxicity, both locally and systemic, interactions of the nanoparticles with other biochemical molecules inside the body in such a way that they have adverse effects on the health of the recipients, the potential of forming cancerous tumors, accumulation of the insoluble particles inside other tissues and organs, and in addition, not being able to be clear from the body tissues. Need to improve the ability to deliver therapeutic molecules selectively and precisely to the chosen target cells, so there is no distribution to any other part of the body. That will be an ideal delivery system to bring these particles to where they're needed, but not have them distributed in other parts of the human body. Always the additional knowledge and understanding of the detailed underlying mechanism is always most important, because it can become the basis on which new solutions can be found and the potential for moving this field in new promising directions. I should add at this point that nanoparticles have found great applications in delivering chemical compounds that can kill cancerous cells by preparing specific particles with the loaded chemical compounds, drugs which kill cells, and by targeting the delivery of those particles at the tumor site has had great potential and has been a major important application of nanoparticles and in nanomedicine. This, of course, aspect is a little bit outside the scope of our interest in the biomaterials field. Additional concerns of biomedical engineers are related to technological and manufacturing issues. The production of these novel materials and particles requires novel manufacturing processes, and so far what we know about these processes, they are difficult to control. The synthesis of nanoparticles for applications in nanomedicine must be optimized so that the process always gives reliably the same product all the time. The produced either biomaterials or particles have to be clean, sterile, without anything that will cause fever, allergies, or other inflammatory reactions once it is delivered inside the human body. And in order to assure the sterile, acceptable in the medical environment product, these particular rules and regulations must be observed at all levels of the manufacturing process, starting with the raw materials, processed water, and all other levels of the manufacturing procedure. Once produced, the nanomaterials must not change during handling and storage. We have to keep in mind that because we're dealing with products which will go inside the body of humans, the technological and manufacturing aspects have to be according to appropriate regulations for marketing authorization and approval by the appropriate agencies. Science-based standards are needed to bridge the current gap between manufacturing and what the regulatory agencies require. We need to enhance the reproducibility at a high scale. For example, the results that I showed you from the research in my laboratories are the ones that require perhaps small samples of materials and a few perhaps drops of one chemical compound and a few thousand cells. In order to manufacture, I mean by that, a product which will reach the market and be used by our colleagues in the healthcare fields, we have to have large quantities of these special products produced. Therefore, the processes which we are currently developing in the laboratories have to be scaled up for that kind of marketable production level. Last but not least, we have to have new materials and new pertinent manufacturing methods for nanoparticles and nanomedicine products. Biomedical engineers are designing, formulating, and producing products which are used by our colleagues in the healthcare fields to treat patients in the clinics and the hospitals. Those colleagues have their own concerns and needs regarding the biomaterials products, especially the nanostructure ones. Some of those concerns are similar to the ones I expressed as the concerns of biomedical engineers, starting of course with the toxicity at the local and systemic level, interactions with other biochemical molecules inside the human body, which of course cause all kinds of unwanted adverse health effects. Accumulation of the insoluble particles inside the human tissues and organs, such as the liver, spleen, and lymph nodes, etc., are of major concern of our colleagues in medicine, because if these nanoparticles are affecting the function of major organs in the body, such as the liver and the spleen, for example, then all kinds of unwanted and problematic complications may arise for the patient, and they may counterindicate and perhaps may cause adverse results for the recipient patients. One way to optimize the performance of the novel products is to optimize the performance, for example, the clearance rate and where the waste products go and as they're removed from the human body. Such outcomes will need new analytical measurements, which could be related to toxicology, to determine the appropriate dose for delivery, and in addition, assessment of associate risk for all of these new applications. Like any other new field, nanostructural materials and nanotechnology overall are developing fields which need to establish appropriate and sound scientific fundamentals, standards, and methodologies for all aspects: research, manufacturing, and clinical applications. In all these endeavors, the paramount important aspect is that of safety, to produce and manufacture and deliver safe and effective nanomaterials and nanoparticles for clinical use by our colleagues in the healthcare field. Up to recently, the endeavors related to nanostructural materials and nanotechnology have been limited at laboratories, let's say at universities, where the scale of the effort and the number of products, etc., is rather limited. What is needed eventually is to address and bring about the translation of these new methodologies and products to the level where a large number of the products can be put together, reach the market, and have an impact on the needs of the patients in the clinics and the hospitals. Similar to any other product which is going to be used in the human body, nanotechnology and nanostructural biomaterials need to meet the requirements which are imposed on such products by regulatory agencies in various countries. For example, such regulations have been established and we need to abide by them in the United States of America, the European Union, countries in Asia, and in Brazil. Last but not least is another aspect which I didn't touch so far, and this is to determine the potential risk and threats and to avoid them as far as the environment is concerned. People are concerned by the production of all these particles and the degradable nanostructural materials. The question is, once these byproducts are released effectively and successfully from the human body, where are they going to go? Are they going to go down in the ground, in the water supply, in the rivers, in the oceans? Will they have detrimental effects on the life of various organisms in the rivers and in the sea? Or will they affect the plants which are grown in the soil, then eaten by the animals, and then the animals meet eaten by humans, there is a whole cycle of potential effects which are risks and threats that cannot be avoided. They need to be addressed, they need to be resolved properly in order to have the nanostructure materials and nanotechnology products achieve the potential that they are able to achieve. They have great promise, but all of these issues regarding safety and efficacy need to be addressed successfully. My opinion is that there is a future for nanostructure materials as implants and for nanotechnology applications in medicine, as long as the potential health-related risks are recognized, addressed, and resolved successfully. In that respect, a lot of good work needs to be done by biomedical engineers in many fields, including that of the biomaterials one. I will like to take this moment to acknowledge the contributions of former graduate students of mine, Ms. Anastasia McManus, Dr. David Puleo, and Dr. Thomas Webster, whose research results I showed in the slides that I presented to you as results from my laboratory. In addition, I would like to recognize the collaboration of colleagues at Albany Medical College and at Rensselaer Polytechnic Institute. Last but not least, I would like to acknowledge the Nanophase Technologies company, which provided Nanophase alumina and titanium powders, which we used in the respective studies which involves ceramic materials. This is the end of lecture 18, Cell Interactions with Nanostructure Materials.